Today we're going to talk about DNA, and I want to start off by talking about just a brief introduction on how the structure of DNA was sort of discovered. In a twist of events, the picture you'll often see is this one here, famous picture with this guy James Watson and Francis Crick, who were given credit along with a couple other people, and they won the Nobel Prize later on for their discovery of the structure of DNA in 1953. And it's an interesting story because they were working on other projects, and they sort of took the ideas of a couple other people and put those together, and were able to come up with this. You can read my lecture notes for more detail on that, and I'll just leave it at that for now. I want to talk about, first of all, when we talk about DNA or RNA, when we talk about these nucleotides, or talk about these nucleic acids, nucleic acids are made up of these smaller monomers called nucleotides. So a monomer is a single unit, that should be an N. And if you put a bunch of monomers together over and over again, you get a polymer. So a polymer is a big molecule made up of a repetition of things, and in this case the monomers can vary. They can be different parts, but they all contain, when we talk about a nucleic acid, it's a long chain of these nucleotides, and all nucleotides have essentially these three parts: a sugar, a phosphate group, and a nitrogenous base. So in the case, we're going to talk about two of them today, DNA and RNA. DNA stands for deoxyribose nucleic acid. So here's deoxyribose, that's the sugar. And in the case of RNA, RNA is ribose nucleic acid that you see there. So difference in the sugar. A couple other differences we'll mention as we go. So ribose and deoxyribose. So that's the sugar. They also have a phosphate group, not shown in this picture yet. I'll show you in a minute. And then they have a nitrogenous base. So those three parts. Okay. Here's another picture showing you a nucleotide. Here's my sugar here. Here's my nitrogenous base. Just shown as a square here, and here's my phosphate group. And what's going to happen is we're going to take this carbohydrate, this sugar here. So the sugar is the carbohydrate, and what's going to happen is they're going to be numbered. We're going to number these carbons as we go. So what happens is, if I go to this picture here and show you what's going to happen when we put the DNA together, we will see that there's going to be what we call the 5' end and a 3' end. And so DNA, which typically comes as a double-stranded molecule most of the time, there are exceptions to that, but we won't deal with those in Bio 1 here. That molecule on one side is flipped upside down. So one side is 5' to 3', but the other side of it is flipped over. So you notice that here we have 5', but on this side we have the 3' side. So what's going to happen is we're going to number these carbohydrates. And so if I go back to this picture here, here's my carbon. All right, so this is number 1 there, number 2, number 3, number 4, number 5. So there's my carbons. And so what we're going to find is that if I go number these once again here, if I start over here on this one here, that's number 1, that's number 2, that's number 3, number 4, number 5. So I'm going to be connecting, as you'll see. So that's 1, that's 2, that's 3, that's 4, that's 5. So we call it 5', 3', because what happens is on this side up here, the 5', so 5', the number 5 carbon, is connected to the phosphate group. And so as I build this structure this way, my next one's going to connect, because they connect to this phosphate background, to the 5' end on one side. And then on the other one, I'm going to connect to this carbon right here, the 3' carbon. So that's if you look at this one here, that's number 1, that's number 2, that's number 3. So what happens is although we can number the carbons 1 through 5, we're going to connect one nucleotide to another nucleotide. So that's one nucleotide there. And here's another nucleotide there. We're going to connect them to the 5' carbon and the 3' carbon. So what happens is one side becomes what we call 5' because that's where the 5' is on the top molecule, and on the other side facing downward is 3'. So if you notice this, that if you look at this particular one here, it goes 5' to 3'. This one's upside down. So notice that if you look at the oxygen there, for example, notice how that one's turned down, facing downward like that. So this is my 5' end on this side because that's my 5' carbon, and that's my 3' carbon. Okay, and that's going to be important later on when we start looking at how we copy this molecule. Okay, this is another way that they show it. So I'll kind of show you this here. This sort of puts it all together in different models. So we have 3 different models here. We have the typical double helix model here, where in blue, in this blue here, this is the phosphate in sugar. And the reason they sort of do that, because what's really important here is the nitrogenous base. So what we find is that the nitrogenous base in DNA, adenine will always pair up with thymine, and guanine will always pair up with cytosine. And the sequence of these ends up being important because this is going to be the genetic code that allows me to code for proteins. That's what we're going to talk about coming up later on. So what happens is the sequence of these, there's 4 different nucleotides. There's adenine, guanine, cytosine, and thymine. And adenine and thymine, notice adenine has these 2 sort of rings and thymine has 1 ring. So the 2 rings are called purines, and those are adenine and guanine. And the single rings are called pyrimidines. So I always remember the longer word is the smaller ring. So we have the purines and we have the pyrimidines. And so thymine and cytosine are my 2 pyrimidines. And what you notice is that the molecule of DNA, when they first were thinking about this, they thought, well, maybe the purines pair up with each other. But notice that how they form these hydrogen bonds. If you measure DNA, it turns out it has these really nice even measurements here. So the width of it sort of stays the same. It doesn't go out and in and out and in. So it stays sort of uniform in its width. And that's how one of the reasons they started coming up with the idea that maybe they paired up like this. Plus they noticed that whenever you measured the amount of guanine, it equaled the amount of cytosine. So if you had a piece of DNA that was 30% guanine, then that same piece of DNA turned out to be 30% cytosine. So the G and the C always paired up, and same thing was true with the A and the T. So for example, you have 30% and 30%, that's 60%. That leaves you 40% left, which could be divided into 20% and 20% of adenine and thymine, for example. So you could figure out the percentages you had of each of those like that. So that's my DNA molecule. Again, it's a double helix molecule with my phosphate and sugars like that. And then what changes, really, what makes one nucleotide different from another is the nitrogenous base. And again, adenine's always pairing up with thymine, and guanine's always pairing up with cytosine. So what we're ready for now is, once again, here's my DNA molecule that you see here. What's going to happen is when this cell, the DNA, first of all, the DNA is the genetic code. And what's important about the genetic code is the genetic code tells you how to make proteins. So ultimately, what a cell can do or not do is based on its ability to make proteins. Because again, proteins, mainly enzymes, control chemical reactions. So to control the chemical reactions of the cell, you need to be able to control the ability to make proteins and certain kinds of proteins. So what we're going to talk about now, two distinct processes. The first one we're going to do today, or right now at least, is talk about how you might take the DNA, and if you had one cell and you wanted to take that one cell and make it into two cells, one of the things you would have to do is copy all of the DNA. Okay, so that is called DNA replication. That's the process of copying all the DNA. Now, since this is a non-majors Bio 1 course, I'm going to skip a lot of the other details and just talk about really a couple of the enzymes involved here. So there's other enzymes that help uncoil this DNA and so forth. We're going to skip those here, and we're going to just talk about these two enzymes here. So what ends up happening is my DNA, I have my DNA molecule here. The original DNA is in blue. Okay, and if I want to take one cell and copy that cell and to have two cells that can then divide and become four cells, I need to copy the blueprints of this genetic code. So what I'm going to do is I have this enzyme called DNA polymerase. And DNA polymerase will hook DNA nucleotides together, but it can only travel in the 5' to 3' direction. So again, remember, that is based on the sugar. I was telling you how we number the sugars. And so in terms of the new strand, so my new strand, as I build my new strand, I can only go from the 5' to the 3' direction. And so there's my DNA polymerase molecule. And you can notice here that I have a C paired up with a G and a C paired up with a G. And so I have this adenine here. So I'm just waiting for the thymine nucleotide to come in here, and then my DNA polymerase will hook that together, and it will keep going on and connecting them as I travel up that. Okay, going from 5' to 3'. Okay, so my 5' to 3' traveling in this direction like that, relative to the new strand that I'm building. Okay, on this side, you'll notice once again DNA polymerase can only go from 5' to 3'. So on this, another DNA polymerase molecule will bind here and will travel this way. Okay, so once again, DNA polymerase can only travel this way. On this side, which is what we call the leading strand, there's no issue because I just keep going on as this unwinds. But the problem on this side of the DNA is I'm doing this piece, and as I copy this piece and move this direction, this has not been copied yet. Okay? That has not been copied yet. So what has to happen then is I have to connect up here with another DNA polymerase molecule and travel along like that. Okay, so 5'. Don't worry about this right now because those are designed to label the blue one. Okay, so if you think about this is the new strand, and that's what we need to pay attention to. You don't need to really worry about what the blue strand is. But so what's going to happen is my other DNA polymerase molecule is going to connect up here, and it's going to travel along and connect that. Okay, DNA polymerase. But once it gets here, I end up with this piece and I have this piece. So I have to hook those two together. And there's a different enzyme that'll hook those two together, and that's DNA ligase. So what happens on the leading strand is I copy the whole thing as I go, and no problem. But on this other side, where I do it in pieces, we call that the lagging strand. The lagging strand gives me what we call Okazaki fragments. It gives me a piece here, and it gives me another piece here. And so to hook the Okazaki fragments together, I use another enzyme called DNA ligase that'll put those two together. So when we're all done, we end up with a copy of the DNA in which on both sides half of the DNA is new, and that's in blue is the old DNA. And so we got half of the DNA is the original DNA or the old DNA, and then we have that's new, that's the orange that's being produced here. So we have half and half on both sides. That's called the semi-conservative model. Semi-conservative because you got half new and half old on both sides. Okay, and again, that's DNA replication. You use DNA replication when you're trying to take one cell, copy everything in that cell, and divide it into make two cells that are identical.